Welcome everyone to Wednesday Night at the Lab. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. I also work for UW Extension and Cooperative Extension, and on behalf of those folks and our other co-organizers, Wisconsin Public Television, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night, 50 times a year. Tonight it's my pleasure to welcome back to Wednesday Night at the Lab Professor William Sugden. He works with the McArdle Lab for Cancer Research. This is the fifth in a series of talks by McArdle researchers. We're commemorating the 75th anniversary of the founding of the McArdle Lab. Professor Sugden was born in Queens, New York City, got his undergraduate degree at Harvard, got his PhD at Cold Spring Harbor, then postdoc at the Karolinska Institute in Stockholm, and then he came here to UW-Madison. Tonight, he's going to be talking to us about understanding Burkitt's lymphoma from its discovery to working towards its cure. This is the second talk that Professor Sugden has given in this series. Please join me in welcoming Bill Sugden back to Wednesday Night at the Lab. Thank you.
Thank you, Tom. What I'd like to do this evening is to point out that Burkitt's lymphoma is caused by a virus, Epstein-Barr virus, and in particular, tell you how we know that. And I want to do that because it's just an amazing story, particularly in its early phase, of people dedicated to cancer research and what a few people were able to do. It's a lovely story. Along the way, I want to address what is cancer, how does a series of questions lead to understanding a particular cancer, and how can we use this understanding to develop therapies for a specific cancer? The story of Burkitt's lymphoma actually begins a long time ago, far away, in a different continent, in Eastern Africa, about 60 years ago. And fittingly, it begins with Denis Burkitt. So Burkitt was trained as a surgeon in Ireland and in England just before and during World War II. And by the mid-1950s, as a mission doctor, he was in Kampala, Uganda. I met Denis Burkitt. I didn't know him well, but I read a lot of his papers and I read a lot about him. And from all of that, I have the impression that he was an amazingly perceptive person whose perceptions allowed him to see things that a lot of us wouldn't be able to see. So I'm going to describe some of that to you too. In 1957, in a hospital in Kampala, a colleague introduced him to a child who had massive growths on both sides of his jaw, upper and lower jaws. He had never seen anything like this, nor had his colleague. He examined the youngster, recognized there was nothing he could do to help that child, and went on about his surgeries. So in general, he had more than 600 surgeries each year in Kampala and in rural clinics surrounding Kampala. And that could have been the end of this story. But several weeks later, at one of those rural clinics, he looked outside and saw another child that presented exactly the same way. Massive growths on both sides of the jaw, upper and lower jaw too. And this was more than a curiosity. This was something he wanted to understand. He examined the youngster. He brought the youngster to Kampala to examine him with his mother, and recognized that this was something that he had not seen before. And he wanted to address two kinds of questions. In particular, what kind of disease did it represent, this new growth that he had found? For example, could it be the response to a parasitic or even a bacterial infection? And did this disease represent a new entity, one that had just entered the population, so it wouldn't have been seen before? To address the first question, he set about just examining youngsters that he found around Kampala. And it's amazing because quite rapidly, he found 36 cases that presented the same way, large growths, both sides of the jaw, upper and lower jaws. But he also very rapidly realized that for most of these children, there were additional large growths elsewhere in their body, in internal organs, stomach, kidney, ovaries, for example. And he reasoned it was quite unlikely that a response to a bacterial infection, a parasitic infection, would give rise to these multiple growths at distant sites in the body. He asked 2 colleagues who were pathologists to examine the cells in biopsies from the same patient, from the jaw and from internal organs. And when they did, they concluded that the tissue was the same. The cells in each of those distant sites was similar, that it was likely to represent a malignant outgrowth of blood cells, to be a lymphoma. To address that second question, Burkitt went to a nearby hospital that was the oldest hospital in East Africa. It had been formed by missionaries in 1897. And he went back and he read their records, just to find out if there were examples prior to that time, prior to the 1957, in which children had presented with this kind of what they thought now was a lymphoma. And there were many examples. There were pictures drawn by hand of children from 1910, 1920 that presented the same way. What those earlier physicians had failed to see is that in these children who had these massive growths around their jaws, they also had growths on internal organs, and they had misdiagnosed this lymphoma that clearly existed long before 1957. So Denis Burkitt recognized that this growth was a cancer because it shared this defining characteristic. It's a disease in which cancer is a disease in which a single cell type proliferates inappropriately and can migrate to abnormal sites in the body, colonize them, and continue to proliferate. And if you think back now what Burkitt had found, growths in the jaws, but growths in internal organs too, the same kind of cell at each of those sites. That's characteristic of a cancer. Now let me just ask you to think for a moment. The year is 1957. You've identified this new form of cancer, newly identified form of cancer, this lymphoma. You have very limited resources, but you'd like to be able to help the kids have this cancer. What would you do? Can you imagine what he did? I couldn't. I would not have done what he did. But what he did turned out to be extremely influential in driving forward our understanding of Burkitt's lymphoma. What he did is to make the decision he was going to map carefully within Africa where children were found that had this tumor. Where did the tumor occur? And he did so in 2 ways. First, he wrote a grant back to people in England for 25 pounds. So in 1957, 25 pounds was about $75. And with those 25 pounds, he had printed 1,000 leaflets. On those leaflets, he described and depicted children with Burkitt's lymphoma, and he sent those leaflets to 1,000 hospitals, rural dispensaries, rural clinics throughout Africa, and asked physicians in them, if you've ever encountered a youngster that presents this way, please tell me. Tell me everything you can about that patient. And from that initial survey, which you've got to admit is an incredibly small amount funded by a small amount of money, he learned that this was the most common cancer in all of Africa, that it was particularly common from 10 degrees north of the equator to 10 degrees south of the equator, and then ran further south along the eastern seaboard of the continent. And he referred to this as the lymphoma belt. The second thing he did is to write a second grant, this time for 750 pounds. Again, it was funded, fortunately. And with that funding, he decided to map the boundaries explicitly of where you found this tumor, and I'm going to call it Burkitt's lymphoma from this time on, where you found patients with Burkitt's lymphoma and where you didn't. So with that 750 pounds, he bought a second-hand Ford station wagon, got 2 colleagues to go with him, and they drove 10,000 miles, largely on non-existent roads, visited 8 countries, 57 hospitals, and interviewed physicians in them all, and interviewed physicians at rural clinics along the way too, inquiring had they encountered youngsters with what we are calling today Burkitt's lymphoma. And from this long 10,000-mile safari, Burkitt and his colleagues learned 2 more things. This lymphoma was common to regions in which the minimum average temperature exceeded 60 degrees Fahrenheit. It was common in regions in which the annual rainfall exceeded 20 inches per year. Now, I challenge you, with that information, what more would you know? I wouldn't know something, but he was surrounded by people who were experts in tropical medicine. And what they immediately could see, which I couldn't see, is that these characteristics are shared by, in fact, a number of infectious diseases in Africa. And what brings those infectious diseases together, what they have in common, is that they're all caused by agents that are transmitted by mosquitoes. So, for example, Anopheles mosquitoes, which carry the protozoan malaria, live with these characteristics, in regions of these characteristics. And in fact, in the gray is shown where Dennis Burkitt had identified Burkitt lymphoma patients, and in the black lines running across that gray area is where malaria was endemic throughout Africa. They overlap almost perfectly. So from this survey, from these insights, Dennis Burkitt made two hypotheses. The first was that perhaps Burkitt's lymphoma is caused by something transmitted by mosquitoes, for example, a virus. That was his first hypothesis. The second hypothesis was that perhaps malaria, which overlaps so well with the incidence of Burkitt's lymphoma, contributes to the cause of Burkitt's lymphoma. The first hypothesis, that there was an insect-borne agent, really turned out largely to be wrong. But that hypothesis was so influential in driving forward our understanding of Burkitt's lymphoma that it was really quite important. And the second hypothesis, that malaria might be linked causally to Burkitt's lymphoma, turns out largely to be true, and I'll come back to that at the end of the talk. The story of understanding Burkitt's lymphoma now shifts to England, to London in particular. But before we go there, I want to give you one more example of Dennis Burkitt's contributions to Burkitt's lymphoma. He wanted to treat those children. What he knew at that time, so this is 1961, 1962, is that in the States, companies were making compounds, molecules that were used as chemotherapeutic agents. They were being tested as such. He also knew that he couldn't conceivably afford to buy those compounds to treat his children. But he knew that in this country, in the States, the standard of care for almost everybody who had cancer at that time was radiation therapy, so that people would first receive radiation therapy and then be tested with these new compounds. Burkitt not only lacked the funds to buy the compounds, he lacked the funds to buy any equipment for radiation therapy, so none of his children could be treated that way. So he wrote the manufacturers of these compounds and said, look, if you give them to me, I'll test them on my children, and there'll be no problems with confounding effects of radiation therapy. They don't have it. And the company said, fine, we'll give them to you. He was the most successful chemotherapist in the world at that time. 20% of his kids survived. And the reason that happened is because Burkitt's lymphoma, then and now, is the fastest growing tumor in people. And as such, it's incredibly sensitive to chemotherapy. So not only was he instrumental in identifying this new or newly identified tumor, he was also instrumental in developing treatments for it. So now let me take you back from Africa to London, when in 1961, Dennis Burkitt, who was on vacation, gave a talk. And the title of that talk in London was The Commonest Children's Cancer in Tropical Africa, a Hitherto Unrecognized Syndrome. In the audience of that talk was Tony Epstein. Epstein is a pathologist who was trained in part in New York City at the Rockefeller University with George Palade to use a new kind of experimental approach in biology, and that was to use an electron microscope. When Epstein returned to London, he decided to use the microscope, the electron microscope, to study viruses. Viruses, as you know, cause lots of diseases in people, but they're way too small to be seen in a light microscope. They're about 1/10 to 1/100th of the size of the smallest thing you can see in a light microscope. So the electron microscope, which can resolve much smaller structures, was ideal to study viruses. And in particular, Epstein wanted to study viruses that cause cancers in animals because it was quite controversial then whether or not a virus could cause a cancer in an animal. And surely there was no evidence that it could cause a cancer in us. So after he heard that Burkitt hypothesized that Burkitt's lymphoma might be caused by a viral infection, he decided to work on Burkitt's lymphoma and search for such a virus. At the end of the talk, he went up to Dennis Burkitt and said, "Can you send me live biopsies from your patients?" And Burkitt said, "Of course." And that was now possible because there was a plane connection between Kampala and London. What Epstein had to do then was to take cells from these biopsies and grow them so that one cell would give 2, would give 4, would give 8, propagate those cells in culture, and then examine them in the electron microscope. And that's what he set out to do. And for 3 years, with 2 colleagues, Yvonne Barr and Bart Ah-Chung, he tried to do so. And for 22 biopsies that were sent by this plane, he failed. They failed. They couldn't get the cells to grow. Nobody had gotten this kind of lymphoma to grow at all in culture. The 23rd shipment was delayed by fog. The plane couldn't land in London. It landed somewhere further north. Several days later, the sample made it to the lab in London. Those samples were solid blocks of tissue suspended in some sort of liquid. And when the 23rd sample came, they looked at the liquid and it was all cloudy. So they looked in the light microscope to see what the cloud was composed of, and it was composed of cells. The cells were growing. We now know that Burkitt lymphoma cells grow best in suspension, but prior to that chance encounter, the tissue had always been grown as a block, and that way Burkitt lymphoma cells just don't grow. So now Epstein could propagate these cells and eventually examine them in the light microscope, and let me show you what he saw. That's what he saw. So this micrograph is a testament to Epstein's tenacity for 3 years of failure and sticking it out and continuing to try to grow those cells. But it's also an example of really luck preparing someone to take advantage of chance. Because in fact, what you're looking at are virus particles seen in the electron microscope, Epstein-Barr virus particles. But the cells that Epstein was growing very rarely ever allowed these to be formed. In fact, about 1% of some cultures would exhibit these particles. And he was really lucky. Of course, he worked like mad to find it, but he was really lucky to observe them. Now let me show you what he was seeing. If I can get this to be, well, if you look at the lower signal, it looks like a hexagon, 6-sided. And what you're looking through is a cut across a virus particle. And that particle is actually icosahedral in 3 dimensions. But when you cut across it, it looks like a hexagon. And if you look again at the lower left, it's dense in the middle. And that density reflects DNA. That's electron dense. That's how it's observed in the electron microscope. And if you look at some of the upper images, what you'll see is there is a faint ring around them. That's a membrane that encapsulates those virus particles. And for people who are experienced at looking at these sort of images, they'll say, "Ah, that's a herpes virus." So he knew what it was. At least he knew in what family of viruses it was. And in those days, we knew that herpes simplex causes cold sores. We knew that varicella zoster virus, another herpes virus, causes chickenpox. And so the question arose, is this a known herpes virus, or was there an unknown herpes virus associated with a small fraction of these Burkitt lymphoma cells? To answer that, Epstein set out to collaborate with a number of people. And in particular, he collaborated with the Henleys in Philadelphia. So Werner and Gertrude Henley were quite well known for a kind of studies called serology, where they studied viruses in infected cells. And in particular, they had studied flu and mumps. When Epstein approached them and told them about his identification of a virus, a herpes virus that might be in Burkitt's lymphomas, they were keen to help him. So let me explain what they would do. Some of you may know, maybe not all of you know, that in our bloodstream we make proteins, antibodies. That's part of our immune response. And these antibodies will bind specifically to virus particles and to the protein constituents of a virus particle, and ideally they inactivate it. That's part of our immune response when we get infected with a virus. And in fact, that antibody response can be thought of as a kind of immunologic memory. Because if I was infected with Epstein-Barr virus, 55 years ago, which I was, I probably still have, I know I do, have antibodies that detect Epstein-Barr virus in my blood. That's the virus that he found. So what the Henleys could do is to take antibodies from many patients, bind them to a fluorescent dye, take these colored antibodies now, and bind them to cells in culture to ask if those cells were making the virus particles. That's the kind of experiment they did. They isolated antibodies from 17 children with Burkitt's lymphoma, coupled them with a dye, then added those antibodies to the cells that Epstein had given them, in which 1% of them exhibited virus particles. And this is what they saw. Most of those cells are reddish. They have no antibodies associated with them. They're stained so you can see the cells in the first place. A few of them are green-yellow. That's the color of the fluorescent dye. That told the Henleys that 1% of those cells were being recognized by the antibodies from the patients. In fact, that children with Burkitt's lymphoma mount an immune response to this virus. The Henleys had antibodies from lots of other sources, ones that were characterized to identify herpes simplex or varicella zoster. Those antibodies did not react with 1% of these cells, from which they could conclude that this was a new virus, Epstein-Barr virus, we now call it, a new member of the herpes virus family that was found in 1% of the Burkitt lymphoma cells. That was very exciting, too, as you might imagine. The Henleys were very thorough scientists. They didn't just look at antibodies from Burkitt lymphoma patients. They took antibodies from many donors, healthy people, people with other kinds of diseases, and they tried them in these sort of assay. And here comes the surprise. When they did that, they found that many people had antibodies to this 1% of the cells, to the virus particles in 1% of these cells. How could that be? That was truly puzzling. A solution to the puzzle came serendipitously in their own lab when one of the people who had been carrying out these experiments, who had used her own antibodies as a negative control, they did not stain these virus particles in 1% of the cells, came down with infectious mononucleosis. She stayed home for a few weeks. When she was better, she came back to the lab. She drew her blood. She used her own antibodies again as a negative control, but it wasn't negative. Now she had antibodies that reacted with virus particles in 1% of these cells, from which the Henleys inferred, and we now know, that Epstein-Barr virus causes infectious mononucleosis. We know also that a lot of us are infected with this virus without getting any disease. In fact, probably almost everybody in the audience, at least 90% of you and me, are already infected with EBV, and we already have it in us, and we have it in us all our lives. So, by the early 1970s, we knew that Burkitt's lymphoma is a common childhood lymphoma whose distribution overlaps that of malaria in Africa. We know that Burkitt's lymphoma cells contain Epstein-Barr virus in some form, and that Epstein-Barr virus infects most people and probably causes infectious mononucleosis. That was what we understood in the 1970s. What I want to do now is to narrow the focus of this talk and describe work that we've done in Madison in the last 40 years. I want to do that so I can describe to you the logic behind the experiments we've used that lead us to look for therapies for Burkitt's lymphoma in a very particular way. But I want to admit immediately to you that with this narrow focus, I'm ignoring the vast majority of all the research that has been done both on Burkitt's lymphoma and on Epstein-Barr virus during these last 40 years. But I'll come back to it, a tiny part of that research at the end of the talk. In 1973, I went to the Karolinska Institute in Stockholm, Sweden, to work with George and Eva Klein. George and Eva Klein collaborated with Tony Epstein and with the Henleys. And in fact, they were instrumental in demonstrating that Epstein-Barr virus is also causally associated with another human cancer, nasopharyngeal carcinoma, which is common in southern China. I went there to learn about Epstein-Barr virus, and by 1975, I'd learned pretty much what I could, and I came to Madison and took a faculty position at McArdle Laboratories. I wanted to answer lots of questions about Epstein-Barr virus, but I'm going to describe two in detail that we've pursued. One is, what does EBV do to the cell it infects? And the other, is EBV maintained in these infected cells? Recall that in Burkitt's experiments, in the Henleys studies, really only a small fraction of Burkitt lymphoma biopsy cells displayed any evidence of being associated with this new virus, Epstein-Barr virus. What was the virus doing? How did it in fact contribute to Burkitt's lymphoma? What does it do to infected cells? To address this question, we have to know what kind of cell it infects. And by 1975, when I came here, it was evident that the kind of cell that makes up Burkitt lymphoma cells is a B lymphocyte. Those are the cells in our bloodstream that make antibodies. It was also possible by 1975 that the infected cell in infectious mononucleosis could be a B cell. That really wasn't shown until 1982, but it was possible. And probably more importantly, John Pope, a researcher in Australia, had shown that he could infect B lymphocytes, I'm going to call them B cells for a moment, or it's easier, B cells with Epstein-Barr virus. So we set out to generate a quantitative assay to study the infection of B cells drawn from your blood or my blood with Epstein-Barr virus. Let me describe the assay. It's actually quite simple and it's quite lovely. So on the left, you see a tissue culture dish that's actually filled with agarose. And agarose you can just think of as sloppy jello. And in that sloppy jello, we could embed B lymphocytes, B cells that we take from somebody's blood. And then we could just watch them in that sloppy jello. Or we could expose those cells to Epstein-Barr virus and then plate them in the sloppy jello and follow them over time. These experiments would take on the order of 5 or 6 weeks. And in the left dish, you saw nothing. The B cells, in the absence of EBV, did nothing but die. They didn't divide, they just sat there and then died. And they died with a half-life of about half would die every 24 to 36 hours. In the right culture dish, for those cells exposed to EBV, after 4 or 5 weeks, what you would begin to see is macroscopic colonies. You could hold it up to one of these lights and you'd see cloudy areas. And you could pick those colonies and put them into cell culture and they would grow. And they look like something like the little bunch of cells shown below of that Petri dish. And so then we could characterize the infection and ask what was happening. We could show that one particle of EBV is sufficient to induce and maintain proliferation of infected B cells, and that those infected B cells would divide. One would go to 2, 2 to 4, 4 to 8, 8 to 16, etc., for 40 or 50 generations. And sometimes they would go on and divide in ad infinitum. They were immortalized. And all I mean by that is if you fed nutrients to these cells, they would continue to divide and divide. To illustrate that, let me give you a number. If you take one B cell and infect it with EBV and allow it to propagate for 50 generations, and if you could harvest all the cells at the end of that 50 generations and you weighed them, they would weigh about 1 ton from one cell. And what that should illustrate for you is the ability of this virus to induce and maintain proliferation could be a hallmark of its contributions to tumor formation. And that's a thought that clearly has driven us. And you'll see it's likely to be correct. Is EBV in those proliferating cells? Is a small fragment in them? Is none in them? You can surely imagine that the virus would go in and have some sort of genetic switch, turn the cell on to divide, and then be lost. So we wanted to know whether EBV DNA was maintained in these proliferating cells, whether all of it was maintained, whether a small fragment of it was maintained, and if it was maintained, how. To describe for you how we've answered these questions, I've got to introduce you to a technique that was just generated about 1976, 77 when we were doing these experiments. It's a little technical, but bear with me. It's called Southern blotting, and it's named after Ed Southern, who developed that technique. And in the center of this image, you can see a representation of DNA, a double-stranded helical structure. And you can think of each strand of that DNA as being Watson and Crick. And those two strands are held together. Watson holds on to Crick, Crick holds on to Watson by virtue of base pairing. So in the left, you can see that the left strand has an A, T, C, and G, and the right strand has a C, G, A, T as I read up. A hybridizes or binds to T, T binds to A, C to G, G to C. That's how the double helix is held together. And then if you read from the top down, T, A, G, C, that's the genetic code that underlies us. You take 1,000 or maybe 10,000 of those nucleotides and you get a sequence of one gene, and all our DNA is made up of is a bunch of genes which can be translated to make proteins, and we're a bunch of proteins, roughly speaking. What we wanted to know, we couldn't sequence the DNA, we wanted to know whether all of the EBV genome, all of its DNA is in these cells, these tumor cells, or not. How could we answer that? Again, we couldn't sequence it. EBV DNA is very big, but it's tiny relative to the DNA in one of your cells. If you take the DNA out of a nucleus and you stretch it out, it's about 2 yards long. If you take the DNA out of those cells or out of an EBV virus particle and you measure its length, it's about as long as your hair, a single hair, is wide. That makes a big DNA, but it's tiny relative to the DNA in our cells. How can we characterize it? Well, there are enzymes, proteins, that will cleave DNA, and they were available then at a specific sequence. So that enzyme, BAM, cuts every time it sees the sequence GGATCC, and it cuts both strands, because you can see it's GGATCC in one direction on one strand, and in the opposite direction on the other strand, it is GGATCC. Every time those 6 nucleotides are encountered by that enzyme, it cuts the DNA. So in the diagram shown below, you can see it cuts the DNA 3 times, generating a large and a small piece, and you can separate those pieces in a gel electrophoretically. The big pieces run more slowly, so A is on the top, B is on the bottom. We could do the same thing now with DNA that we isolate from Epstein-Barr virus particles, with cells that we infected with purified EBV particles in cell culture, and most importantly, we could take Burkitt lymphoma biopsies, isolate DNA from those biopsies that were frozen. We had never done anything with them other than they were sent to us as frozen samples, purify the DNAs, digest with that enzyme, BAM, separate all the fragments in the gel as shown on the right, and then detect what's pure EBV DNA, no cell DNA, because again, Watson binds Crick. So if we purified DNA from an EBV particle, made it radioactive, then Watson from that labeled DNA would bind Crick on that filter, and we could detect that radioactivity with X-ray film. And that's the Southern blot you're seeing. And if you look at that Southern blot, you're not going to be able easily to say whether lane 1 is different from lane 2, is different from lane 3. They're intensities. It's not a perfect blot, but that's a long time ago. But the patterns are the same. And in particular, if you look, there's a heavy band labeled 2.1. It's heavy because that represents a repeated sequence cut by BAM 8 or 10 times in the EBV genome. And you'll see they all have it. It's all intense. So from these experiments and the similarity of the patterns separated from purified viral DNA, from cells infected in vitro, from Burkitt lymphoma biopsy cells, we could conclude that all of EBV DNA is present in those tumors. How's it maintained? Those tumor cells undergo 10, 20, 30, 50 generations, doublings. How is the virus distributed to those daughter cells? It has to be synthesized. How is it synthesized? That's the question we focused on, and if you'll bear with me, I think I can tell you why we focused on it. So what did we do? Well, Chris Kittner, a graduate student, set out to begin understanding how EBV DNA was maintained in these proliferating cells with the following experiment. And this would have been about 1980, 1981. Again, across the center of this slide, you can see all of the fragments generated when you cut linear DNA with the BAM enzyme. EBV DNA in that virus particle is a linear double-stranded DNA. Herpes simplex DNA in a herpes simplex virus particle is a linear double-stranded DNA. All herpes viruses maintain the DNA in the virus particle as a linear molecule. What Chris set out to do is to ask, could he identify the ends of this linear molecule? And he was able to do so. And you can see up there, they're in green. They're labeled the end fragment. They are the ends of that linear DNA. Then he could ask what happens to them. You know, if there's only a fragment of EBV DNA in a cell, then one end might be gone. But what he found is not only were the end fragments there, they were coupled together. And in fact, EBV DNA was maintained intact in these cells as a circle. We call that circle a plasmid, and human cells don't normally have plasmid, circular DNAs, in their nucleus. This was really quite surprising. This then led to the question, how does this circle, which don't normally find plasmids in human cells, how is this plasmid DNA maintained in these proliferating cells? And again, by this time you might say, why is he talking about all this minutia? So let me say why I'm doing it. The hope behind the narrow focus of our research is this. If we know how EBV DNA is maintained in tumors, and if we know EBV helps to sustain these tumors, then we can hope to treat the tumors by forcing the loss of EBDNA from those tumor cells. That's our hope. And that's what's driven the rest of our research in the past and, in fact, in some ways, to this day. So let me tell you how we have at least answered the first two if clauses and what we're doing for the last hope. John Yates was a postdoc. He set out to understand mechanistically how these plasmids derived from EBV were maintained in cells. And he had two hypotheses. The first was that somewhere in this circle there had to be a sequence of DNA that allowed its synthesis to begin. This DNA had to be synthesized before cells divided so it could get passed on to daughter cells. So there should be a sequence somewhere in that DNA that allows it to start synthesis, a sequence in the DNA. He wanted to find that sequence. The second is, since you don't normally find plasmids in human cell nuclei, probably EBV was encoding one or more genes that allowed this origin, this site at which DNA synthesis initiates, to function. So he wanted to find viral genes that acted at this hypothetical origin. And he did both. Let me show you how he did it. Again, you can see EBV DNA now arrayed as a circle, as you would find it inside a Burkitt lymphoma cell. When you cut it with BAM enzyme, you get all of those fragments. What John did is take each fragment and make a mini plasmid out of it, and then introduce it in the cell, which had EBV intact plasmids in them already. We reasoned that if a cell had an intact plasmid, it had to make all of the functions necessary for that plasmid to replicate, to be passed on to daughter cells. So John put each one of these small fragments alone into cells that already had EBV plasmids and asked, could any one of them now behave itself as a mini plasmid? One and only one did, the BAMC fragment, from which he concluded that somewhere in the BAMC fragment was a site at which DNA synthesis initiated. To ask or answer the question, could there be genes encoded by EBV that allow that mini plasmid to be synthesized? He took each one of those fragments again and then put one at a time into cells that had no other EBV in it. And you can think of this as taking EBV, which is a bunch of about 100 genes, and then breaking them up one gene at a time and putting one gene at a time into a cell that had no other viral genes. Then into that cell, he could introduce the mini plasmid, the BAMC fragment, and ask, could it replicate as a plasmid? And after hundreds of attempts, John found that the BAMK fragment provided the only functions EBV provides for BAMC to replicate as a plasmid. Everything else comes from the cell. For us, this was a great step forward. It was after 3 years of work where 4 of us worked together, but John was the person who spearheaded this research. Let me illustrate schematically what he had found. There are 2 genetic elements required for the plasmid replication of EBV. In the middle, in that line with those bars, is the sequence drawn out schematically that represents the BAM-C fragment and its origin of synthesis we call OREP, for origin of plasmid replication. What we now know is the BAM-K fragment encodes the only gene product EBV needs for that BAM-C to replicate as a plasmid. It's a protein we call the Epstein-Barr virus nuclear antigen 1, or EBNA1, and it binds to all those white bars in OREP. When it binds to the origin, it allows DNA synthesis to initiate by recruiting lots of machinery from the cell. When it binds to the white bars in the black and white pattern, what it's doing is holding the plasmid next to adjacent to sites in human chromosomes, and that helps to bring it to daughter cells when the chromosomes go to daughter cells. We've studied OREP and EBNA1 a lot. And in those studies, one thing that we really wanted to be able to do was to see OREP in a live cell and watch it be duplicated when it was synthesized and ask how is it distributed to daughter cells. That would be a nice experiment. And we actually did that experiment. Really, Aska Nambo did that experiment. And she learned something that we did not expect we would learn. And that piece of knowledge allowed us to say we know that EBV provides Burkitt lymphomas a selective advantage in order to be maintained as a plasmid. So I want to show you Aska's experiments. So on the left, you can see one cell with one dot, I hope. And that's before the cell makes any DNA. So it's just at what we call the beginning of the cell cycle. And below, in a cartoon, you can see the intensity that Aska measured of that one dot, and it's 143 arbitrary units. Cells synthesize DNA at a given time. We call that the synthetic phase, or S phase. And after S phase, you can see that that signal becomes more intense, and you can read the intensity down below. It goes from 143 to 250 arbitrary units. What's happened is that the EBV plasmid has been duplicated, but it doesn't separate so far from itself that we can resolve that in a fluorescent microscope. So what are you looking at? What you're looking at is Aska took OREP plasmids and introduced additional sequences to them. These sequences bind an irrelevant protein that's fluorescent. So you can tag the viral DNA with this fluorescent signal and observe it in a fluorescence microscope. And now that's what you're seeing. The dot on the left represents one plasmid, and because you can measure the intensity of that fluorescent signal, the second dot is twice as intense, approximately. We know it got duplicated. This is all because, again, the plasmid has added to it additional sequences that bind a fluorescent protein. After the synthetic phase, cells divide. Now look what happens to the plasmid when they divide. Each daughter cell gets one, and you can see that the intensity goes down by half. And that's exactly what we would have guessed would happen, and it did, 26 out of 31 times. The 5 times on the right is what we did not guess, did not expect, and they turned out to be really informative. Again, in G1, before the synthetic phase, you can see a cell with one signal, and its intensity in this cartoon is about 177. After the synthetic phase, the intensity does not go up. If anything, it goes down. You can follow that one cell when it gives rise to 2 daughter cells and look at the signals in the 2 daughter cells, and only one cell has a signal, and its intensity has dropped slightly. From which we have to conclude that in 5 of these examples, the EBV DNA was not synthesized. Now you might ask, so what? Well, what this really tells us, and we've confirmed this in many different cell types, that about 16% of the time, EBV DNA fails to be synthesized in proliferating cells. Again, so what? Well, just think about it. Start with any number of molecules you would like in a cell. In most of these cells, usually there are about 10. Aska had to look for cells that had only 1 so she could follow one signal at a time. But most of the time, there are about 10. If the cell divides, for there to remain 10, each daughter cell has to get, on average, 10. But if you fail to synthesize some of the DNAs, maybe you're only going to get 9 and 9, and eventually 8 and 8, etc. If there are defects of this magnitude in synthesis, what will happen is as the cells divide, they're going to lose EBV DNA, and eventually those cells will have no EBV DNA. Okay? Except when you take out Burkitt lymphoma biopsies and you look in those cells, every cell has EBV plasmids in it. How can that be? And that was the insight that Oska's experiments allowed us to draw. So some EBV plasmid DNAs fail to be synthesized each synthetic phase, leading to their decrease in average number per cell each cell cycle. The implication of this finding is that EBV will be lost from any population of proliferating cells unless it affords cells a growth or survival advantage so that those that have it outgrow those cells that lose it. All Burkitt lymphoma cells have EBV DNA in them. They're all plasmids. We conclude those plasmids must be providing those tumor cells one or more selective advantages so that the cells that actually have lost EBV, and they're going to be there, just have passed away. They've died, they're outgrown, we don't see them. We want to know what the selective advantage is that EBV affords these Burkitt lymphoma tumor cells, and Dave Verrady, a graduate student, set out to answer that. What is the selective advantage? The way he did it is really based on something you've already heard. I've said that EBNA1, the one protein encoded by EBV, is essential for EBV plasmids to be synthesized and distributed to daughter cells. Dave made a derivative of this protein that could block the function of the normal protein. I'll call it an inhibitor. He could then introduce that inhibitor into a wide variety of Burkitt lymphoma cells and ask what happens. What happens is they all lose EBV DNA. They all lose the EBV plasmids. Then what happens? So in this plot on the y-axis, you just see the number of doublings of the cells he's following. 0, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, etc. And by 10 doublings, one cell will give rise to about 1,000. And on the x-axis, you see the number of days that he followed these cells. And the black line shows the cells in which this inhibitor is not expressed, and they're growing perfectly. And the red line that's dotted shows what happens when the inhibitor is expressed. Those cells lose EBV plasmids, they cease to proliferate, and they die. EBV is providing, if you think about it, a massive selective advantage to Burkitt lymphoma cells. It allows them to survive. It also helps them to grow, we know now, but it allows them to survive. That was a very marvelous day, we understood, too. So we now know EBV induces and maintains proliferation of infected B cells. EBV DNA is maintained as a plasmid in these cells. EBV must afford Burkitt's lymphoma cells advantages to be maintained in them. And when EBV plasmids are lost from them, Burkitt's lymphoma cells die. It would be incredibly nice if we could take Dave Verrady's approach and take a derivative of EBNA1 and introduce it into Burkitt lymphoma cells in Burkitt lymphoma patients and treat them successfully, but we can't. That's an experiment you can do with isolated cells in a lab. You cannot do it in people, surely not in a population in rural Africa. But we would love to build on the finding that if you inhibit EBNA1, you can cure cells of EBV DNA plasmids, and that forces the cells in general to die. So how can we do that? Let me describe the efforts of Yen Lam and Mitch Hayes in the lab, who are now screening for small molecules that will do the same thing and can be used in patients that Dave Verrady's derivative of EBNA1 did. Let me explain how they're doing this. We know from the research of a number of groups that this EBNA1 protein has to come together as a dimer to function. It can only bind to DNA in the BAM-C fragment when it's a dimer. It can only carry out any of its known functions when it's a dimer. So what Yen and Mitch have done is to take half of firefly luciferase. That's the molecule in fireflies that emits light when it encounters substrate, luciferin and ATP. But they took a half, which has no enzymatic activity, and fused it to EBNA1. They took the other half and fused it to another molecule of EBNA1. No light's emitted unless EBNA1 forms a dimer and brings those two halves of luciferase together. And if you inhibit that dimerization, you inhibit light emission. This is a very rapid assay that you can now look for inhibitors of the dimerization of EBNA1. You can introduce these fusions into cells, then introduce a compound you want to test, and ask, have you inhibited light emission? So with funding from the Leukemia and Lymphoma Society, with help from Mike Hoffman, who has really begun and directs the small molecule screening facility in the Comprehensive Cancer Center here, Jan and Mitch have screened more than 400,000 compounds. They've screened them in multiple assays. From those 400,000 compounds, they've reduced their workload to about 90, which are candidates for inhibiting EBNA1's dimerization. If any of these molecules actually inhibit the dimer formation specifically, it would serve as a lead to develop a therapy for Burkitt's lymphoma, and that would be marvelous. So I want to end with two additional thoughts. One is I want to bring you all back now to this large body of work that has been done on Burkitt's lymphoma and on Epstein-Barr virus for the last 40 years that I haven't touched on at all. And I want to do so just to briefly address a question which I imagine some of you have been thinking about. And that question is, why do many of us get infectious mononucleosis? I had it. Anybody else had an infectious mono in the group? Several of us have. Why do many of us get infectious mononucleosis while few of us develop Burkitt's lymphoma? The answer to this question, in part, comes from Dennis Burkitt, but it comes from a number of people who have addressed this question in England, in Australia, and in Sweden, other places too, but the main groups have been England, Australia, and Sweden that I know of. What they've found is that chronic malaria, and that's what you find in Central Africa, suppresses a patient's immune response. This suppression allows EBV-infected cells to escape the immune response. They continue to proliferate in these children, for example. And when they do proliferate, they acquire mutations. Those are just mistakes in DNA synthesis of their own genes, not EBV's genes, but their own genes. And that those mutations can promote tumorigenesis. So Dennis Burkitt's hypothesis turns out really to be correct, the second one. EBV and malaria are cofactors in the cause of Burkitt's lymphoma. And that really explains why in this country, something on the order of 1 in 200,000 children might develop Burkitt's lymphoma, and in Central Africa, it's more like 1 in 10,000. The last point I want to leave you with is a picture of an extended family, our extended family. And in the left, in the back, you see Mitch, and in the front, you see Yen. Those are the people who are carrying forward this screen of now more than 400,000 compounds. And we surely hope we're going to find a specific one that we can then use to develop better, more potent inhibitors of EBNA1's dimerization. When you look at this picture, you'll be able to tell probably that we come from at least 5 countries, 3 continents, and if you count me, we span much more than 50 years of age differential. If we don't succeed in finding that inhibitor that we're looking for, what I can promise you is that this younger generation will. Thank you.